Hello students, now we will learn about meiosis. Meiosis is also called as reductional division. Meiosis means formation of 4 number of haploid daughter cells from one single diploid or haploid mother cell. Because there are different types of meiosis like zygotic meiosis, sporic meiosis, etc. So meiosis, also called reduction division. Meiosis results in formation of 4 haploid cells, right? Haploid daughter cells. Now, what is difference between diploid and haploid? Diploid means that particular cell will be having 2 sets of chromosomes here. For suppose, one chromosome is said as one set, other chromosome is said as other set. So in case of diploid cell, if this is a cell, it will be having one set, means 2 in number of chromosomes. Haploid cell, if this is a cell, it is having only one set of chromosomes. So here each chromosome is considered as one set. Actually set means 2 in number, but here we are considering one chromosome as one set. The term is given by Farmer and Moore. Meiosis is discovered by Van Benden. Here, the cell that undergoes meiosis is called as meiocyte. There are different types of meiosis, that is gametic meiosis, zygotic meiosis and sporic meiosis. Different types of meiosis are, first one, gametic meiosis. Second one, sporic meiosis. Third one, zygotic meiosis. Now what is gametic meiosis? Gametic meiosis occurs from a diploid cell which is having 2 sets of chromosomes, and here the obtained gametes will be having haploid number of chromosomes. Gametic meiosis means diploid. Here 2 sets of chromosomes are represented as 2n, one set of chromosome is represented as n. So we are writing meiosis means diploid cell undergoing meiosis, giving out haploid gametes. Such type of means gamete formation happens mostly in higher organisms. For suppose fungus we can take, animal cell we can take. So here zygote is also diploid, but it is not undergoing here in gametic type of meiosis. Example, in fungus, in higher animal cells or higher organisms. In In case of gametic meiosis, zygote is not undergoing meiosis. In case of zygotic meiosis. Zygotic meiosis is seen in haplontic life cycle. In Chlamydomonas, in lower algae, etc. Here, zygote undergoes meiosis to produce zygospores. Now, we'll write the ploidy. The zygote that is undergoing meiosis is of 2n, but after undergoing meiosis or reduction division, the produced offsprings, which are 4 in number, are haploid in number. Each daughter cell is haploid in number, means having half the number of chromosomes. Next, sporic meiosis. Sporic meiosis is production of haploid spores from a diploid mother cell. Sporic type of meiosis seen in haploid phase of haplodiplontic life cycle. Examples in which this type of sporic meiosis occurs. Example in pteridophytes. So these are types of meiosis. Hello students, now we will learn about the process of meiosis, how meiosis happens, what are different stages in meiosis. So, meiosis, it is happening in 2 steps. It is happening in 2 steps. That is, first step or stage is meiosis 1. Second one is meiosis 2. Meiosis 2 is equal to mitotic cell division. It is equal to mitosis type of cell division. Meiosis, this process includes 4 steps, that are prophase 1, metaphase 1, anaphase 1, telophase 1. This is karyokinesis. Now, meiosis 2, it includes prophase 2, metaphase 2, anaphase 2, telophase 2. After this karyokinesis, up to here it is karyokinesis. After karyokinesis, if you give a numbering as A and this one as B, cytokinesis happens. Now, here also, this is cytokinesis 1, this is cytokinesis, means division of cytoplasm, kinesis 2. There is a time period between cytokinesis 1 and meiosis 2. Starting of prophase, means some ATP, some energy might have been utilized here. So again, cell has to get ready for meiosis. This time period we are giving a term here as interkinesis. Before mitosis or meiosis or M phase, we have given a term like interphase for the time period when cell is getting or preparing itself for mitosis or meiosis. Same likewise here after cytokinesis 1, before starting of prophase 2, cell will be having some time period to get prepared for itself for meiosis 2. That time period is called as interkinesis. Interkinesis. Now, interkinesis does not have S phase, does not include, but in case of interphase we will see S phase, G1, S phase, G2 phases. But here in case of interkinesis there is no S phase. Coming to meiosis 1, the first one, prophase 1. It is again divided into 5 different steps, that is leptotene, zygotene, pachytene, diplotene, diakinesis. So during karyokinesis, all these steps happen. After cytokinesis 1, interkinesis takes place. Then cell enters into meiosis 2. That it will start prophase 2. After that, cytokinesis 2 happens. Now we will see in detail about all these stages and substages. First one, prophase 1. Under prophase 1, we said that it is of there are 5 different substages. First one is leptotene. Under leptotene, what is happening? If this is an animal cell, so we'll write meiosis in animal cell. If we take this one as a cell, and this is nucleus, initially what happens, there will be chromatin and that has to get condensed into thread-like chromosomes. Initially it will be, chromatin will be like a highly coiled long thread-like structure. Since we are telling it is an animal cell, meiosis happening in animal cell, so there will be centrioles. There will be, we'll write here. We cannot initially take that centrioles are at opposite poles. So this is nucleus, so this will be nucleolus. These will be centrioles. What is leptotene root word meaning? It means that formation of long thread-like structures. Here, lepto means thin, also called as leptonema or leptonema. Here, nema means thread-like. So thread-like structures are formed in leptotene substage of prophase 1. So totally leptotene means the substage in which thin thread-like chromosomes are formed. By condensation of chromatin. Next one. So this is leptotene or leptonema stage. So here this is of male gamete, from male gamete. This long chromatin will be from female gamete. So, when two such male and female gametes are releasing chromosomes into one single area or cell, then such chromosomes which have come nearby are called as homologous chromosomes. In meiosis, the chromosomes that are paired are called homologous chromosomes. What are homologous chromosomes? Homologous chromosomes means two chromosomes that is means two chromosomes that is one from male gamete, next second chromosome from female gamete, and both are paired. So now they will be present inside the egg nucleus. So such chromosomes are called as homologous chromosomes. Here those homologous chromosomes we have mentioned as long thread-like. Now they will be changing their shape and structure a little bit now. Next one, zygotene, which is also called as zygonema. So after leptotene, the next stage is zygotene or zygonema. Here zygonema means a pair. A pair-like structure is formed from these thread-like chromosomes. Now, formation of pair from these thread-like chromosomes. Here nema means thread-like. In case of zygotene or substage zygonema, maternal means from mother. And paternal that is from father. Chromosomes come side by side, to each other and forms a pair called homologous pair. We will draw the structure regarding that. This is a cell, animal cell. This is nuclear membrane. Nuclear membrane is not yet disappeared. Centrioles. Next, this will be nucleus. Nucleolus. Think that this is from mother, this chromosome, and other chromosome from father will represent with other color. This is from father chromosome. Both are called homologous pair. Since they are arranged side by side to each other. These will be centrioles. We'll give some differences. So in case of zygotene, there are some another terms. We are seeing these two number of chromosomes arranged side by side just as a pair. Now, so when two number of chromosomes are being appeared side by side as a pair, such condition is called bivalent. And the chromosomes are called bivalent chromosomes. As a pair, now these chromosomes are said as bivalents. So if a question has come, when is bivalent stage of chromosomes seen? The answer will be in zygotene, the bivalence of chromosomes are seen. Now, arrangement of two different types of chromosomes side by side as a pair. That arrangement is called as synapsis. Means such type of chromosomes must be able to later exchange their genetic material. Such type of arrangement or means of forming into a pair-like structure is called as synapsis. The condition of arrangement of two types of homologous chromosomes that later can exchange genetic material is called synapsis. So we have seen here two different terms. One is bivalent, arrangement of two chromosomes as a pair. Now, those paired chromosomes must be able to later exchange their genetic material. Such type or condition is called synapsis. Each chromosome will be having two chromatids, 4 arms. So, when they are rotating inside the nucleus, we at one stage, at some point, we can see 4 chromatids. So, this zygotene stage is also said to be containing tetrad forms. Because of having 4 chromatids, this zygotene stage is said to contain tetrad. Tetrad means, tetra means 4 in number. What is 4 in number here? We are seeing only 2 chromosomes, but each chromosome will be having 2 chromatids. So totally 4 number of chromatids are there. So we are giving a term like tetrad. So this stage is also given with another term, that is when 2 homologous chromosomes are paired side by side, that appears like a bouquet. So this is also called as bouquet stage also. Zygotene also said as bouquet stage. So this is about zygotene or zygonema, and next the before one leptotene or leptonema. Next is pachytene or pachytene. This is third substage of prophase 1. So we will use C. Pachy means thick. Nema means thread-like. Under Pachytene, you draw a cell, nuclear membrane or nucleus, nucleolus means still by this stage nucleolus is present. Nucleolus. Clearly in Pachytene stage, in Zygotene we have seen chromosomes like this, homologous pairs. This is of father and this is of mother. In Zygotene we have seen like this. The same chromosomes from Zygotene now will show four number of chromatids very clearly. From mother, now the same homologous chromosome. Now from father. Centrioles now. They tend to move to opposite poles, but not yet completed. In case of Pachytene or Pachynema, crossing over takes place. We also write crossing over starts here. We can treat this one also as an MCQ. So crossing over starts in Pachytene. So we can write this is an MCQ. That is starting of crossing over. So between what types of structures does this crossing over? If a chromosome is like this is one chromosome, this is other chromosome. Here this arm is there and this arm of other chromosome is there. These are said as, this one and this one, two red color one are said as sister. This means complete chromatid, sister chromatids or sister arms. Now, but one chromatid of one chromosome, other chromatid of father chromosome, this one, we can take from female chromatid, from male chromatid. These both are said as non-sister chromatids. But two chromatids from same chromosome are said as sister chromatids, non-sister chromatids. I'll draw here the structure clearly. If this is the chromosome from mother, think that this is from father. We said that this part is one chromatid, this part is other chromatid. In the same way, in father chromosome, this is one chromatid, this is another chromatid. So this is 1, 2, 1, 2 chromatids. This chromatid are called sister chromatids. Same, this and this chromatids are called sister chromatids. But this chromatid from one female chromosome and from father chromosome, this one, these together are called as non-sister chromatids. Now, in case of Pachytene stage, crossing over happens between these two non-sister chromatids. Non-sister chromatids cross over each other at a point called chiasmata. Now, this part is called chiasmata. If you want to draw that one. This is from female gamete, this is from male gamete. Now, if this is one chromatid, this is other chromatid, they are crossing at one point. This point is called chiasmata. So they are crossing over each other at this point is chiasmata. Will be having an outer layer called pellicle. Protein layer called pellicle. Now, these chromatids have attached at one place to exchange the genetic material. So each chromatid will be having outermost layer that is digested by one enzyme called recombinase. So if an MCQ has come, what is the type of enzyme that is involved during meiosis? And that too, if they specifically ask during prophase 1 or during Pachytene, then we can easily recall. But all over meiosis, one enzyme specifically involves in digesting the pellicle of the chromosome during crossing over, that is recombinase. Enzyme named recombinase is involved to digest pellicle at chiasmata region. So, after digestion of the outermost layers, then the inside genetic material of this chromosome and this chromosome will be forming into a network-like, means inside the chromosome or chromatids, the genetic material exchange will take place. That forms into a network-like or a complex-like structure called as synaptonemal complex. Exchange of genetic material takes place, forming a synaptonemal complex. We can also say a network or network-like structure. Network-like structure. So exchange of genetic material takes place, and here we are seeing new term, that is synaptonemal complex 1, chiasmata, enzyme recombinase, crossing over, starting. So 1, 2, 3, 4. 4 important points. Next is Diplotene or diplonema. This is fourth substage of prophase 1. So what are changes happening after Pachytene in Diplotene stage? Now, here we have seen nuclear membrane, and cell organelles also will be present, and centrioles also will be present. Now, centrioles starts moving to opposite poles. Nuclear membrane of nucleus, it starts disappearing. Even nucleolus also disappears. Nucleolus and nuclear membrane starts disappearing. Next, if you draw a structure, this is a cell. Nucleolus, now it gets disappeared. Centrioles now starts moving to opposite poles. So these are centrioles. Now, since nuclear membrane and nucleolus are getting disappeared, I am not labeling them. Here, in case of Diplotene, crossing over has started here. By the time of Diplotene, crossing over gets finished. So, crossing over ends in Diplotene. So, crossing over started in Pachytene. Crossing over ends or ended in Diplotene. So, crossing over started in Pachytene. Crossing over ended in Diplotene. So, after ending, then what is happening to the chromosomes? Mother chromosome gets repelled like this. mother chromosome also gets repelled like this. Means crossing over has finished. Now here some genetic material has entered into this chromosome. In case of father chromosome, now, some material of female gamete or female this chromosome has entered here. We can think that this red shaded one in one chromosome and blue color type of chromatic bit in other chromosome are nothing but exchanged parts of chromosome. Now, we will write the same diagram in explanatory form. Non-sister chromatids now repel to each other, undergoes repulsion. Now they undergo repulsion. Centrioles starts moving to opposite poles. So, when they are undergoing repulsion, it means that the synaptonymal complex that is formed now got broken. Synaptonymal complex breaks. We can write these both are non-sister chromatids. Non-sister chromatids. They got repelled with each other. Nucleolus got disappeared now inside the disappearing nuclear membrane. Last one, diakinesis. Diakinesis. This is the last one. Here, this is the area where previously during Pachytene they have undergone crossing over, and at the same time during Diplotene they underwent a repulsion. That area, that chiasmata region, now it will be moved to terminal part of non-sister chromatids. So here again the cell. Now I am not giving out nuclear membrane. Directly we will write about centrioles. Now, mother chromosome. Spindle fibers, they starts forming, and now chromatids, non-sister chromatids, they started undergoing terminalization. Terminalization will be completed and finished here. Now, diakinesis includes terminalization. Sometimes this diakinesis step itself, this substage itself is called as terminalization of meiosis. Means during the meiosis, one terminalization step is included. What it is here, it will be diakinesis. So we have written here diakinesis includes terminalization. We'll write that also of crossover chromatids. non-sister chromatids. Next. But still the non-sister chromatids will remain attached at the tip. Non-sister chromatids remain attached at the tips. Next. Nucleolus and nuclear membrane completely will be disappeared. By now, nucleolus and nuclear membrane I am writing as capital N, capital M, are completely disappeared. Next, centrioles are moved to opposite poles and both centrioles are attached by spindle fibers. Since it is of animals, we are talking about centrioles. If not, we don't talk about centrioles while discussing meiosis in plant cell. So these are spindle fibers. So this is about prophase 1 and all its substages: leptotene, zygotene, pachytene, diplotene and diakinesis. Totally there are 5 different substages included in prophase 1.